Hello everyone. In this lecture, you will learn briefly about the history of microscopes. We will review parts of the microscope, and finally you will learn how to measure objects under the microscope and microscopes, how they are used to study the living environment. The discovery of the lens was linked to the invention of microscope. The art of grinding lenses was developed in 14th century in Italy to make spectacles to improve eyesight. In 1667, Robert Hooke studied various objects with his microscope and published his results in Micrographia. In this publication, he described the structure of the cork and its ability to float. In 1675, a Dutch draper and amateur microscope builder, Anton van Leeuwenhoek, used a simple microscope with only one lens to look at blood, insects, and many other objects. He was first to describe cells and bacteria seen through his very small microscope. Only in 19th century, the microscopes improved and their widespread use to study microorganisms helped understand the microbial life forms. Here's a comical depiction of early microscope. They were crude, large, limited in their capacity to magnify, and very cumbersome to use. Parts of a compound microscope. So let's go over them now. The type of microscope that you will be using throughout your study in microbiology is oil immersion compound microscope. The compound microscope achieves higher levels of magnifications than a stereo or lower power magnification. This will help us view small cells like bacteria with more detail. Compound microscope contains two lenses that cause light rays to bend in a way to produce an enlarged image. A compound microscope consists of structural and optical components. Compound microscope inverts the image and makes it upside down. With increased magnification from 40x to 100x, the object becomes larger and the image becomes smaller. Now let's look at the basic structural components first. Head or the body consists of optical parts in the upper part of the microscope. Arm connects to the base of the microscope, and the head or the stage is connected to the platform. This is where you will place your specimen to view. Finally, base is the support, and it contains the illuminator. Whenever carrying the microscope, you must hold the arm in one hand and place your other hand underneath the base to support it. The optical components of microscope are the lenses. Ocular lens or the eyepiece is the lens through which you look at the specimen and has the standard magnification of 10x. Objective lenses are the primary optical lenses that range from 4x to 100x and typically includes 3 to 5 lenses. Diopter adjustment ring allows for correction of eyesight. Nose piece holds the objective lens. The objective lenses are mounted on a rotating turret. Coarse and fine adjustment knobs are used to focus the microscope. Coarse adjustment knob is used to focus under low power only, and once you move to the higher power, You never ever move the coarse adjustment after that. Use only fine adjustment to focus on your specimen. Aperture is the hole in the stage through which light is transmitted to the stage. Mirror or illuminator directs light up through the specimen. It's located in the base. Condenser is used to collect and focus the light from the illuminator to the specimen. Diaphragm controls how much light can pass through, so you can adjust the diaphragm to make your image brighter or darker. Discs with different sized iris or openings are present in the diaphragm. Magnification is how much you can enlarge the images. Simply, you multiply the strength of two lenses to find the magnification of a microscope. For example, if ocular lens is 10x, the high power objective lens is 45x, then you simply multiply 10x times 45 and you get 450x, which means your image has been enlarged 450 times. Let's go back and look at this image of the microscope. You will get a similar picture and you will have to label the diagram. So you must understand the different parts of the compound microscope. Now let's talk about the important units for microscopes. 1 centimeter, we all know, equals 10 millimeter. And 1 millimeter equals 1000 micrometers. So how much is a micrometer? Micrometer is 1/1000th of a millimeter. So let's look at the ruler. That's your 1 millimeter between the two lines. So imagine there are 1000 microns in that 1 millimeter. That's how tiny micron unit is. So here's a little egg practice for you. Solve these problems to find out the total magnification. Now, what is resolution? Resolution means how sharp and clear you can get the image. Resolution can be increased by adjusting the light. So in your measuring with microscope lab, you will find the diameter of the field of view using the ruler, and I will show you the formula in the next slide. So how do we find field of view? So now microscope, you are looking at very small objects. So how can you find the size of those minute objects? So here is a very clever way. Using a mathematical formula, you can estimate the size of any object that you view under the microscope. So under low power, you will place your ruler, and those are the two lines, the millimeter lines that you saw on the ruler, and you must place those two lines right in the center of your field of view. So that gives you an estimate that in between those two lines is 1 millimeter distance. On the sides, then you approximate that it's 0.25 on each side. Therefore, your diameter of the view is 1.5 millimeter. Now 1.5 times 1000 equals 1500 micrometers. Now this is the formula you can use to calculate the size of any object that you're viewing under your microscope. High power field diameter equals low power field diameter times low power magnification divided by high power magnification. So let's look at the numbers. Let's say you have placed your specimen under 40x. And you need to find what's the size of the individual object. So let's first calculate the diameter under 40x. We know under 10x, you were able to see 1.5 millimeter diameter, which is equal to 1500 micrometers, times the low power magnification, which is 10, and you want to find out the diameter under 40x. So you divide it by 40, which tells you that the diameter now is 400 micrometer. That means your field of view has decreased, but your image has enlarged. It's almost like you focused in. So you have focused in, your image got bigger, and your diameter got smaller. So remember, magnification increases, field of view decreases. They are inversely proportional. Determining the size of an object under a microscope. So now you will use some practice problems, and few things that I have given you, you will view them under your microscope to actually understand the functioning of microscope. So there are other types of microscopes. Dissecting microscope is very low magnification. It ranges between 10x to 20x. And you do see the true image, but it's right side up. Specimen can be alive under the dissecting microscope. You can use tools for dissecting specimens, and you can actually view the tissues and all the other organs. It's binocular, so you can see 3D image. The next is the phase contrast microscope. These use special filters to show the depth of the cells that enables the researchers to view interior details of the living cells. Now, we do not have phase contrast microscopes. We cannot afford them yet. Electron microscopes magnify images up to 200,000 times, and it uses beam of electrons. The sample is placed in the vacuum, and living specimens cannot be viewed. It allows scientists to view the organelles of the cell. So you can imagine how much depth can be seen using electron microscopes. And that's the end of our lecture. Hope you've enjoyed and learned something, and now you will practice all these skills in the lab. Thank you.